Good morning, everybody. Thanks for joining us and being ready this morning. Just a bit of a thank you for last night to the Albury Botanic Gardens and also to Ben Berry for their talks at the gardens down by the Murray River. That was fantastic. And for the pizza oven, the food that we got, that was fantastic from Vinbar as well. Best Western plus Hoveltree, and our sponsors for drinks, Bilson's Beechworth, Bridge Road Brewers and Campbell's Rutherglen Winemakers. It was a great night, so thanks everyone for coming up. Today's, just want a quick announcement around presentations. If you've got presentations later this morning or this afternoon or even tomorrow, please feel free to pop in behind the curtain here and hand over your talk so we can get it loaded. And this morning's session, despite my best efforts yesterday, they've asked me to do another chairing session, so you're stuck with me one more time. I promise this is the last one. So today's theme 2, Seeds and More, natural regeneration, revegetation and specialist propagation techniques. First up, our keynote speaker is Dr. Mark Ooi, who is a plant ecologist and senior research fellow at the University of New South Wales, where he is a member of the Centre for Ecosystem Science in the School of Biological, Earth and Environmental Sciences. He runs a lab focused on fire, plant and seed ecology and is particularly interested in studying the role of seeds in driving demographic processes of plant species. So the keynote address will be Seeds and Plant Species Persistence under Shifting Fire Regimes. Thanks, Mark.
Hello, thanks for coming in early as well. First, I'd just like to acknowledge the traditional custodians of the land where I work and live, which is on Darawal country and in Gadigal land as well. And also nice to be down here in Albury, where it's the southernmost bit of the Wiradjuri. We do work with northeast Wiradjuri looking at cultural burns. So it's nice to sort of do the full span, the expanse. So yeah, today I'll be looking at seeds. So seeds are important, obviously, and I want to look at the ecology of seeds. They're important as a resource for restoration, but also they drive a lot of the population dynamics. So fire drives ecosystems around the world, and you can see from that pretty picture, that's where across the globe there's ecosystems that fire is a main component. And within these ecosystems, there's tens, hundreds of thousands of species, many thousands of which are at risk. Sometimes they're threatened by different elements of the fire itself, shifting fire regimes. We have a responsibility as conservation biologists, as practitioners, and just as people living here, really, we just need to look after these ecosystems as best we can. And the way we need to look at that is to understand the systems and understand how species persist within those systems. And so we think of plants being adapted to fire, but really it's the fire regime. As we know, climate change is having a big impact on around the world, and it's having an impact on fire regimes. So fire regimes are shifting. And we need to understand how these shifts in different elements of the fire regime can affect plant persistence, the component species. So just as a recap for you, most of you will know all this anyway, but fire regime is made up of several key components. These ones listed here are some of the key ones, I think, in relation to plant species. So frequency is how often a place burns, how frequently it burns. Severity and or intensity sort of correlated, how hot that burn can get essentially. Season, when in the year a fire occurs, and extent, just the expanse over which a fire regularly hits. Frequency is by far the most studied element of the fire regime, and a lot of our understanding, a lot of the management is based on particularly immaturity risk. You don't want to burn an area, say, with an obligate cedar too often, too frequently, because you'll get immaturity risk where the seedlings won't be able to mature, produce enough seeds to then replenish the seed bank. So all of these components have an effect though, and some aren't as well studied as fire frequency. So we did a paper recently, this was led by Rachel Nolan out at Western Sydney Uni, and it's a lot in this graph, so I'll just walk through it. So you can see in that part A, in the top left of the figure there, under a fire regime under which species are already adapted to, you get a cycle where you've got unburnt vegetation, the fire will come through, and then there's all these mechanisms that seeds will be released or seeds will germinate to make sure that you've got recruitment into the post-fire environment when you're needed. And so we have canopy species like Banksias, which is that first picture there, post-fire flowering species like the Dorianthes, Gaimea lily there. And then you get seed germination from the soil seed bank where you get a flush of emergence from heat or smoke cued species. And that's a picture of Grevillea kalei, one of Tony All's favourite species up in northern Sydney. So the fire cues these elements, you get seedlings that emerge into the post-fire environment, and then you get a recruitment event, you get time enough for those species, those seedlings to mature, produce plenty of seeds, and then the cycle continues. If you look down now at part B, this is all the potential changes if you change different elements of the fire regime. So the top one there is fire frequency. If you burn something too often, particularly things like obligate cedars, you'll get that immaturity risk that I just spoke about. If you burn at a severity or an intensity that's beyond the sort of thresholds or limitations a species can persist under, you have a chance of knocking down that species as well. And the one the idea we seem to most fit into this idea is canopy-held seed banks and mortality thresholds of the seeds in the cones. Fire season shifts can also have an impact of reducing recruitment, and there's combinations of all those elements. And one sort of hypothesis that was developed a few years ago, Neil Enright and colleagues, about fire interval squeeze. So you're getting more fire weather and more drought that's driving fire. So you get drought pre-fire, drought post-fire, and then more frequent fires on top of that. So it's a general amalgamation of things that can cause a decline. Now, all of these elements, like I said, some of them are quite well studied, but others there's nowhere near as much information. So we can only really guess at what these impacts might be. One thing you can see from all of this is seeds are important. They're one of the key drivers of how well a population will go. And with the 2019-20 fires, which has been the impetus for a lot of this stuff, it's provided a canvas, I guess, of areas that you can study that have been subject to a gradient of severity up to extreme severity fires with other fire histories underlying it. And that, as you'll see in a minute, is important. And so we can potentially use seed traits then, because seeds are such a driver, we can use seed traits to try and understand and improve our predictions or prioritisations on species that might be at risk. So here is one of many maps I think you've seen of the 2019-2020 fires. There's been a few frameworks that have been developed, and Rachel gave a few examples. So one that Rachel Gallagher's led, and Tony All's led another one at the state level. And the idea was to rapidly assess what happened as a result of the impact of these mega fires and these unprecedented fires. And both the keynotes yesterday looking at this stuff, Rachel and Melinda's, they were exceptional examples, I think, of how you can utilise the data that you have at least to prioritise. And so it's not just prioritising where you're putting the limited resources, it's prioritising species that actually need assistance, I guess. So with Melinda's stuff, for example, you're trying to identify those species that actually need say seeding work. Of course, there's going to be a large number of species that will do best just by recovering in situ, perhaps with some other management. Okay, so these are really important because of the resources that we have available and the priorities we need to set. So frameworks such as these are structured around what we do know. So there's things that are included in them like, okay, what happened if we have higher fire frequency or increased fire frequency? The immaturity risk is an issue. So we know that that's well studied. There's a little bit in there about severity of fires, but again, it's wrapped around canopy species and how they might be knocked back. These were always, because of the need of them being rapid, these were always desktop analyses that the idea was to send out people to actually have a look at these priority species. And they've done really an amazing job at highlighting some of these key species at risk. But we want to also try and improve the ability to predict what's potentially at risk. And so I'm going to talk about seeds. Seeds is something I studied for a while. I do love seeds, and I never thought I'd say that out loud, but they're really interesting study sort of life history stage. So yeah, a couple of seed traits that I think are underutilised as far as this predictive part of the ecology of species is, well, a characteristic or a trait, depending on what you want to call it, but seed dormancy. So seed dormancy, there's about 5 seed dormancy classes, and a couple of the classes you can separate out for species that behave quite differently based on these traits. So physically dormant seeds, or PY seeds as they're known in shorthand, are those with hard seed coat, hard-seeded species. Some people know them as acacias, all that P group. And to germinate into the post-fire environment, you need a heat shock. So there's a heat shock from the passage of fire that will heat the soil and then promote germination. Other species are physiologically dormant, and they have a mechanism within the seed itself, and that block is broken down by seasonal changes in temperature, so warm, cold stratification or dry after ripening. The important thing to know about these species is that they usually have a seasonal temperature required for germination, so they'll germinate into a particular season. And to get into the post-fire environment, these are the species that respond to smoke. So they'll have a response that they'll be cued to smoke to know, this is the time to come up post-fire. The other seed trait that I'll be focusing on a bit for this, and I think should come into these predictive things, is a classic one, which is seed size or seed mass. And yeah, we'll go into that in the next couple of slides. So how can we use some of these traits to try and predict what's happening? If we look at season of burn, physiologically dormant species or PD species have that seasonal germination requirement. So we've looked at a few species in our lab at least, mainly around the Rutaceae and the Ericaceae, and like to expand it to a few other families the more we go into it. And a few students, and I'll be popping photos up of various students who have done work on this, and you can see their happy smiling faces. Justin Collette, who finished his PhD with us a year or two ago, worked on Australasia buxifolia, which is a species some of you might know. Bob Mack found it a couple of decades ago, or rediscovered it. And we wanted to see what are the germination mechanisms. So we tried winter, summer, spring temperatures regimes, applied smoke, and we found that you can see from those 2 bars of germination, they're the smoke-treated seeds, and they're only germinating at winter temperatures. So a species like this is obligated to smoke, so it needs smoke, and it's obligated to come up during winter. So that means when you burn at different times, the emergence of that species relatively is going to be delayed depending on when you burn. So this figure that came out a while ago now, you can see if you burn close to the winter emergence season for this species, so say in an autumn burn or a late summer burn even, it's a pretty short time period before you get the seedling flush. Whereas if you burn, say, right at the end of winter, that species has to wait nearly a whole year before you'll get the seedling flush. And in between times, you'll get competition from resprouting and different things. So this delayed emergence can delay maturity. And that, in effect, then means you need to have a longer fire interval to make sure that species persists okay. And the light grey bars in that final figure there, you can see that's the trajectory of maturing or growing plants coming up after a winter burn compared to after a summer burn. So it's a much slower progression to maturity. And so what this example shows is that if we're looking at trying to predict the impacts of something like the 2020 fires, we need to have an understanding of fire history. So what did the particular site hit by a fire by 2019-2020 fires? When was the season of burn in the previous fire? Are there species that have this type of dormancy or this kind of recruitment mechanism that could be affected? Okay, so if we move on to now looking at actually some of the impacts of the 2019-20 fires, Another student, Tom LeBreton, had a paper out recently, his first chapter out of his PhD. And as we know, these fires were extreme, and the scale of the extreme level of them is the thing that really is crazy, because we do get hot spots, we do get really extreme fires or burn areas within fires. That's part of our fire regime at a bigger scale. But we were getting, say on the south coast of New South Wales, we were getting tens of kilometres at extreme severity, which is really mind-blowing. What Tom found in his study was that having fires at this extent and severity, it was overrunning areas, one, that don't burn at all very often, but also overrunning areas that had been burnt relatively recently before. And so, in essence, you're getting a landscape scale increase in fire frequency, so the first element of interval squeeze. And so, as I said before, this provides an interesting canvas to have a look at what's happening to these species and what's the impact more broadly on the ecosystems, the component species as well. So, yeah, so where to from here? Key and novel traits such, I mean, they're not that novel, they've been around for a while, but we've not used them that much for these sorts of frameworks or prioritisation. I think understanding these elements are really important to understand the greater impacts of changing fire regimes. But I just wanted to finish off a little bit, as well as sort of this ecological sort of understanding of seeds. We also work a little bit within the lab group on very applied, trying to improve germination outcomes or outcomes for restoration. So once we actually have our priorities and we've got a species that we know needs some very direct and applied restoration, I just thought I'd give you a little insight into some of the things. So this is work mainly that's been done in collaboration with Miriam Munoz-Rojas, who's a research fellow at UNSW, recently moved back to Spain after 10 years or so. And we've been looking at harnessing cyanobacteria from soil biocrusts and then extract them out, grow them up in the lab, and then actually biopriming seeds before placing them into restoration conditions to see if that improves their germination outcomes. And what we're finding across a range of species, so we've had positive responses from eucalypts, grevilleas, Triodia. A lot of this was done in semi-arid systems for restoration after mining. We found that biopriming with cyanobacteria is has a positive outcome, both in the germination, but mainly on the seedling growth of species that you bioprime them with. One of the outcomes that's been quite interesting is that it was one of those sort of lucky eureka things. We had a highly saline environment that the cyanobacteria was growing in. And we found that salt-sensitive species, if you bioprime them with cyanobacteria, you can actually have really good germination and really good seedling growth in saline areas. So for recovering saline areas, this might be a really nice approach. We're looking at, well, when I say we, Miriam and some of her students now are looking at pelleting this stuff and seeing if there's a similar outcome.
So next up we've got Dr. Nathan Emery. So Nathan is a Restoration Biology Officer at the Royal Botanic Gardens, Sydney, based at the Australian Plant Bank, part of the Australian Institute of Botanical Science. Nathan's research is a combination of plant ecology, seed biology and restoration. And Nathan's talk today is a data framework for predicting seed germination under climate change using R. Thanks, Nathan.
Thanks, Damien. So this morning I'm going to give a bit of a whirlwind overview of a data framework that myself and my co-authors recently developed, which enables us to predict seed germination under climate change. Now, seed germination is a critical and highly sensitive stage of the plant life cycle, and climate change is a key global concern for many of our plant species. The predicted increase in temperature may potentially impact the germination and timing and success of many plant species. Now, when we're looking at the effect of temperature on germination, traditionally we use germination incubators set to seasonal temperatures for that species. So we have one that's reflective of summer, another for winter, and then one that reflects spring, autumn conditions. But the reality is a seed can germinate at any temperature regime in between these seasonal temperatures as well as outside of them. And this is where the thermogradient plate really comes into its own and is a really powerful tool for germination studies. So if you're not familiar with a thermogradient plate, it is essentially an aluminium plate which is heated along one edge and cooled along the other. So it effectively creates kind of like a gridded a gridded cell display of germination where the differences in temperature between those cells are roughly the same. Now, in more modern machines, this heating and cooling element can be flipped to the perpendicular edge, which then enables kind of like a day-night cycle that we can then replicate on the thermogradient plate. For the work that we do with the thermogradient plate, we tend to only use one half of it because the other half, the seeds will experience their hotter temperatures during the dark or the night cycle and the cold during the day, which doesn't really make ecological sense. So why we use this thermogradient plate? Well, we can examine temperatures, the effect of many, many temperature combinations on seed germination. And this is kind of a unique thing for germination biology because we're effectively testing seeds at their current known temperature range as well as potential future temperatures and other temperature combinations that they may not be experiencing. So it got us thinking, well, we should be able to make use of this data to then predict how germination might respond to local temperatures and changes to the local temperatures. And the other main benefit of using a thermogradient plate is that it typically uses fewer seeds than the equivalent number of germination incubators. Now, there's been a historical hesitancy for using thermogradient plates, and usually this is due to the fact that they're quite expensive. You know, if you purchase one thermogradient plate, it's probably, you could get a few germination incubators for the same cost. But for what I'm presenting to you today, I'd argue that their value and their uniqueness sort of justifies that cost. Because of the way that they heat through the aluminium plate at the bottom, we can't actually follow traditional incubator experiments where we might have, say, 4 Petri plates of 25 seeds in each of those. Each Petri plate is our replicate. We can't do that on the thermogradient plate. The Petri dish which is contacting the aluminium plate is going to experience a temperature regime that is different from the plate that's stacked above it, and that's going to increase the more plates you have on it. So we can't use the typical ANOVAs, the GLMs that we tend to use for a lot of our seed biology research. And we have to look to new or alternative modes of replication. And for this, we use the seed as the unit of replication. So here in this diagram, we have 10 seeds on the plate. 2 of them have germinated. So our final germination proportion for that temperature cell with the plate is 0.2. And we've determined that, or we recommend that using at least 10 seeds per plate is kind of the minimum that you want for your thermogradient plate experiment. You could use less seeds per plate, but you will run into some statistical penalties and inaccuracies. Oops, this is more sensitive than a rainforest seed to drying. So we wanted to make something that was globally relevant and extremely user-friendly. And also highly automated. And we turned to the R platform. I know that if you're not familiar with R, it can seem very intimidating, but we wanted to make this as easy and as pain-free as possible. The benefit of R is that it's freely available, it's widely used, and there's a lot of online help that you can try and troubleshoot any problems. So we produced this R workbook, which has a number of scripts within it, which you can download for free using the Zenodo web link at the bottom of the slide there. And each script has a set of instructions to guide you through what it's actually doing and what sort of interaction you need to have with the script in order for it to run. So I'm going to run through a few of these, an overview of these scripts within the workbook to sort of guide you through the process and the framework that we've developed. The first step is to calculate our germination for each temperature cell on the thermogradient plate. Now, to do this, we use the R package called Germination Metrics, which is a really powerful R package. It has the capacity to produce over 40 germination indices. So if you're not just interested in final germination, you can look at things like T50, day of first germination, mean time to germinate, the list goes on. The one main downfall of this package is that it requires you to do consistent interval checks for your germination, which we all know it's just not possible in reality. We all value our weekends. We're not necessarily going to go in and check on our seeds. So the first step was to write a script that fills in the gaps between our checks using generalized additive models. It interpolates what the germination should be between our checkpoints, which enables the execution of this germination metrics package. Okay, so we have our index that we're interested in, and for this talk I'm going to focus on final germination using 3 case study species that we trialled this framework on. So the first thing we do is to view our raw data visually using rasterized quilt plots. These are triangle plots because we're only using half the thermogradient plate. Here we have day temperature on the x-axis, night temperature on the y-axis, and the fill or the z-axis is our final germination. And for Electrion subdentatus on the left, we see that it's getting a higher germination under warmer temperatures. In contrast to that, Calitris bailey in the centre is showing a preference for cooler temperatures for its germination, and the Wollemi pine on the right has showing no real sort of temperature preference for its germination. It's germinating quite consistently across the plate. So we now get into the nuts and bolts of this workflow, which is generalized additive model, which could seem like a very scary term to many of you, and it was to me when I first started investigating this. But this is essentially non-linear model fitting of our germination in response to temperature. We don't expect a linear response of germination to temperature. So as temperature increases, we're not always going to see increases in germination. And to make this as user-friendly as possible, we try to make it completely automated, but there is some user input that's required in it. So we've got a number of potential models that can best fit your data, and the script will run through these and spit out the table there, which basically gives you a number of measures in which to assess your model fit. So here we've provided a number of different ways for checking your model fit. We have the correlation, the R-square factor, which is on the far right-hand side of the table. We also have the RMSE value, which is the root mean squared error value, which is a common way of assessing model fit. And we also have the mean error of the model, which is a non-parametric measure. So here, this table is for Electrion subdentatus. We'd pick model number 4 because it has the highest correlation of all the models, just over 96%, has a low RMSE error value too. So the only input and interaction that we need with this step is to actually cut and paste this model into the next script in order to progress with the workflow. Okay, so we've chosen our best model. The next step is to then visualize that model and look to see how well it fits in with our raw data. So here we have our raw data for our 3 species on top. Here is the model data, and we can see some really good fitting between the raw and the model data for Electrion and Calitris. Whereas for the Wollemi pine, the correlation factor is quite low, and that's generally what we expect to see when we have a species which isn't really showing any particular temperature response. Okay, so we've got our model data. Now comes the really fun stuff, which is to link that germination with the local temperature site. So the user can input a decimal latitude and longitude value for anywhere in the world. Typically, we expect this to be the collection site for your species. And the script will then download monthly mean, minimum, and maximum temperatures for that site using the WorldClim database, which is freely available online. It will then produce a line graph so you can have a look at temperatures at your site, and then it takes this information with the model data and outputs a prediction of monthly germination in the current climate for that species. So here, for this, we're using Electron subdentatus. We see a much higher prediction of germination to occur during summer and spring temperatures. And we can take this a step further and extrapolate to predict germination under future climate scenarios. So the script will download any number of models, any number of future climate models from the WorldClim data set, as well as any number of climate change scenarios. By default, we've set the script to download every single climate model that's available on the WorldClim dataset for the scenario SSP 5.8, which is the most extreme predicted climate scenario for the year 2100. Again, we'll produce a line graph so you can look at the changes in temperature that's predicted between the current and the future. And then we produce another bar chart which shows predicted germination at the site under future climate. Thanks. And when we put these two graphs together, we can start to visually see how germination may change between the current and the future climate. So here for Electron on the left, we're seeing a potential shift away from that summer germination to more of a spring and autumn germination. Calitris baileyii, which you may remember has a preference for that cooler temperatures for germination, it's predicted to retract to have more of its germination basically in winter alone, whereas for the Wollemi pine we're seeing no real change in its germination, but there is an increase in uncertainty under future conditions. So I'm going to quickly run through the limitations and considerations that you need to keep in mind for this approach. Obviously, don't forget about seed dormancy mechanisms. You need to understand how to or what dormancy type your species has before you put on the thermogradient plate. Scarify your seeds if they're physically dormant before they go on. We're talking here about temperature or germination response to temperature. We're not looking at other potential environmental factors that may be important, particularly in the field, things like moisture, disturbance, and soil. We can sort of overcome some of the assumptions associated with replicating at the seed level. This can be done by running your species multiple times on the thermogradient plate and using each run as a blocking factor in your analysis. But really, I just want to highlight that this approach is not actually analysing statistical differences between temperature cells like what we may do between incubator conditions in a lot of previous seed biology research. We're essentially doing nonlinear curve fitting here to fit germination to the temperature curve. As I mentioned before, this typically uses less seeds than the equivalent number of incubators. And if you're not actually interested in the germination response to current and future climate, it's also a really powerful script to be able to identify the optimal temperatures for germinating a species for the highest germination and the most rapid germination, just by using that first script of the germination metrics package and being able to get over 40 different germination intervals without that need for regular interval checking. So this is really a unique way to be able to start exploring the germination niche of species and identifying and potentially prioritising species which may show potential germination failure under future climate conditions. And if you want to know more about the process, there's a paper link I've put down the bottom of this slide for the study, which has just been recently published in Annals of Botany. If you've got some germination data or want to know and see this script in action, come and chat to me. I'm happy to run you through it and to really show you how easy it is. And I'd just like to acknowledge the traditional custodians of the land where this project was conducted, and also to Parks and DPE and the National Arboretum for use of data and material. Thank you.
Thank you, Nathan. That's a really great example of how we can maximise the limited resources we've got, but particularly in regards to the restoration sector and how important these kind of considerations are for seeds that actually can be used and will be able to establish. The next talk we've got is an online one. So we've got Dr. Ganesha Liyanage, who's been a plant conservation scientist at Australian Plant Bank since 2018. So the primary area of her research is seed dormancy and germination, with a focus on advancing conservation of Australian rainforest species, specifically for ex situ seed storage. And the talk today is Understanding Seed Biology to Inform In Situ and Ex Situ Conservation Actions, a case study on Geijera parviflora.
Hi everyone, thanks for joining to listen to my talk today. I'm speaking to you from Camden, New South Wales, which is before I start my talk. Today I'm going to present results of part of a project that I've worked on understanding seed biology of Geijera parviflora to inform in situ and ex situ conservation actions, particularly by looking at seed dormancy and soil seed bank behavior. So why did we choose Geijera parviflora as our study species? Geijera parviflora is commonly known as wilga, sheepbush, or dogwood, and has a wide distribution from dry woodland to dry rainforest. It's ecologically important as a key species in several endangered ecological communities in northwest New South Wales. There is renowned focus on growing drought-tolerant plants like wilga to raise the water table and stabilize soil. Geijera is also identified as a plant that can provide shade and fodder for stock and also is suitable for mine restoration. Occurring over a wide range of soil types, its bushy structure and mass of scented flowers reflect its potential suitability as a streetscape plant. However, difficulties in germination due to dormancy, low seed fill, and limited knowledge on natural recruitment inhibit its uses. Seed dormancy is a character that prevents germination in the absence of dormancy breaking cues. This is a ecologically important factor because this can enable seeds to germinate only when favorable seedling establishment conditions are present. But in terms of conservation, presence of dormancy can be problematic, as it can prevent seed biology research or production of seedlings for restoration purposes. Studies on soil seed bank give the information on how dormancy can change over time and how long seeds can live in soil seed bank. This is critical to understand the natural recruitment strategies of species. So in this project, we studied the dormancy, particularly on dormancy breaking technique and soil seed bank behavior of Geijera parviflora. Now a little about our methods. We collected seeds from 10 mature adult plants and used 7 dormancy break-in treatments. Then compare the effect of treatments on seed germination. The dormancy break-in treatments we used were presence of gibberellic acid, leaching seeds under running water for 24 hours and 48 hours, and germinating them on gibberellic acid incorporated agar. And also we use scarification of seeds and complete seed coat removal using wires and germinating them on water agar, gibberellic acid, incorporated agar, or with crushed seed coats. We also use set of intact seeds along with treated seeds to assess the dormancy and germination. For each treatment, we used 3 replicates of 15 to 20 seeds, and those seeds were incubated in 25-10 degrees Celsius alternating temperatures. This was the most suitable temperature we used from our preliminary studies. This graph shows germination percentages in response to the dormancy break-in treatments that we used. Highest germination was observed when seed coats were fully removed using wires. But presence of GA, gibberellic acid, and crushed seed coats significantly significantly reduce the germination even the seed coats have fully removed. Intact seeds did not show any germination. What we found from this experiment? We did not observe any germination in intact seeds, and this indicates a 100% dormancy in Geijera parviflora seeds at the dispersal. When we look at the graph, we saw that scarification did not increase the germination considerably. That means permeability of seed coat is not the reason that controls germination. Therefore, physical dormancy is not evident in parviflora. We also performed an imbibition test along with this, and it also confirmed the permeability of seed coats. Increase in germination only when seed coat was removed indicates that embryo required to have a clearance to emerge and can't push through the seed coat by itself, which is a characteristic of physiologically dormant species. These results suggest the presence of physiological dormancy in Geijera parviflora. Use of wires to remove seed coat can be considered as the most effective treatment to achieve higher germination in Geijera parviflora. Then we look at the soil seed bank behavior of Geijera parviflora to understand the limitation associated with its natural recruitment. Geijera parviflora is an obligate seeder. Seedling establishment therefore depends on the soil stored dormant seeds. These dormant seeds form the soil seed bank. As most of you will be aware, the soil seed banks develop by accumulation over one or more years of dormant mature seeds. While in the soil, seeds can receive dormancy breaking or germination cues which elevate dormancy and allow germination when conditions for seedling establishment is favorable. Otherwise, seeds can deteriorate or become non-viable over time in soil. Additionally, soil-stored seeds experience different environmental conditions over time within soil, which may change their morphology and or physiology that control germination. So we don't know whether both fresh and soil-stored seeds follow similar mechanisms in dormancy breaking, and how long seeds of Geijera parviflora remain dormant in soil seed bank. So what we did in here, we collected fresh seeds as explained above. Then we stored them in the field exactly where those been collected from originally. And we added 50 seeds into each mesh bag containing river sand and buried those in 3 locations of selected site. 1 bag from each of 3 locations was retrieved after 6 months and 12 months of soil storage to assess dormancy and germination. We assessed the germination of intact and seed coat removed seeds. This graph shows the germination of intact and seed coat removed seeds at each retrieval time. Germination was significantly decreased after 6 and 12 months storage. Interestingly, we observed around 10% germination in intact seeds after 6 months of storage, which indicates an elevation in dormancy within soil seed bank of dormant seeds. Almost all seeds become non-viable from 6 months to 12 months storage time, with most seeds having decayed that time. We noticed some dried out germinants within mesh bags in each retrieval, but did not observe any seedling establishment within 12 months of this experiment. This shows percentage of seed fill at each retrieval time. Seed fill also drastically decreased from 80% at the dispersal to low as around 10% in seeds retrieved after 12 months. So what we learned so far? Soil seed bank behavior experiment reveals a really interesting insight for natural recruitment of Geijera parviflora. These results indicated that 100% dormancy that we observe at the dispersal gradually alleviate within soil seed bank, making seeds available to germinate as soon as they experience the germination cues. Geijera parviflora seeds are short-lived and deplete within 12 months in the soil seed bank. Our results indicated a complete depletion of soil seed bank within 6 to 12 months after dispersal. We did not observe any seedling establishment during this experiment, which is quite unfortunate and indicates those seeds that become non-dormant responded to germination cues. Early seedling establishment stages are critical. Current conditions in the field may not be suitable for those stages. What we can conclude from our results? This knowledge is significant when using Geijera parviflora in horticulture, agriculture, restoration and ex situ conservation. Seed germination can be achieved in laboratory or nursery conditions by full seed coat removal. And it is essential to perform regular seed collections to maintain a viable seed collection. Maintaining favorable conditions for early seedling stages are critical when producing plants. These conditions could be temperature, watering, etc. And further research on seedling establishment can help to understand these conditions. I would like to end this presentation by thanking the Australian Flora Foundation and the Rainforest Seed Conservation Project at the Australian Plant Bank for their funding support to this research, and my research collaborators for their support throughout the experiments. I also would like to thank Justin and Veronica for their assistance in the field. Thank you.
Thank you, Ganesha. Thank you, Ganesha. It's a great example of how seed collections are living collections, and we have to keep managing them actively. They can't just go in the bank and be forgotten about. Our next talk is Andie Geurin, who's come to talk about there are giraffes in the seed orchard again. I'm intrigued by this one. Andie's currently coordinating the Regent Honeyeater project, a revegetation project based around Benalla in northeast Victoria, which has operated as a not-for-profit for 25 years. Thanks, Andie.
So I'm Andie Geurin, Project Coordinator for the Regent Honeyeater project. We're a not-for-profit based in central Victoria, around Benalla, Glenrowan, Lurg and the Winton wetlands. And I do want to acknowledge our funders up front. I really do appreciate it. This is the rough region in Victoria that we are in. We do lots of revegetation, protection and restoration of remnants, especially with box ironbark. We do landscape scale connectivity for a range of threatened species and EVCs. And over 25 years, we've enhanced over 2,000 hectares. In 2015, a seed orchard was set up, or SPA, or Seed Production Area. This was after a presentation by Linda Broadhurst on genetics, seedling health and seed viable populations. The seed orchard was set up on Tony and Anne Mort's property, and here they are in 2-year-old hot bushes. And unbeknown to us at the time, there was a nest of chicks just next door. So in 2 years, you can have a quick result. Our aim was to have as many provinces as we could, often 15 or more for each species. And many of our provinces have very few plants remaining, single-digit numbers. We travel some distance to access the gene pools of remnants with larger numbers. And the phrase we use is, we travel as far as we need to access genetically healthy populations. We have proportional representation within the seed orchard, so small numbers only have small numbers represented. And we have roo and rabbit-proof fencing, but not giraffe-proof. Now, I need to out myself. This is photoshopped. I'm a practitioner, not a scientist, so please accept any misuse of terminology. And I'm putting forward some concerns and looking for guidance. With giraffes, there's been a recent proposal to break up single species of giraffes into a minimum of 4 separate species. Splitting a single species up into more species can have a profound effect on the number of animals that are left in each of the new species. There were 117,000 giraffes in the wild, and what was thought to be one species. Now, with 4 or more, one species, the northern giraffe, has less than 6,000 animals, so it's rarer than the black rhino and is suddenly endangered, and is further divided into 3 subspecies. Some of the species can be identified by different skin patterns, and now that it's pointed out, it's pretty obvious. Zoo breeding programs, it's now assumed that successful programs were dealing with animals from the same species, and less successful or unsuccessful were dealing with animals from new species. We have giraffes in our seed orchard. Keep in mind, I'm talking revegetation, not restoration, not the very specific species that require a particular niche, many of which I love intimately, but instead those often referred to as the bread and butter species, once common, once widespread species, which I argue are currently rare. Just a few. There's varnish wattle, Eutaxia, clustered everlasting, and gold dust wattle. Varnish wattle, we have long been aware of very different leaf forms and phyllate forms. We have limited remnants in our region. Recent studies by Maslin and Murphy suggest that taxonomic revision of the Acacia verniciflua and leprosa complex. Initially, I was, oh my God, I'm so confused, but the new key is helpful. They suggest that Acacia verniciflua is perplexingly variable, particularly in phyllate shape and size, and this variation does not appear to follow any obvious geographic patterns. However, scanning with electron micrographs of phyllate resin glands and head cell numbers could help confirm. As a practitioner, while this is fantastic information, I just won't get to it. So it would appear currently we probably have variants, perhaps not different species, in our SPA. Then there is Eutaxia. We have 4 growth form types that can be repeatedly found throughout our region. Often 2 of these forms can be found in clumps right up next to each other, and they seem to stay true to type when grown by seed. We have the prostrate form. That was my boot. Oh, that is my boot. We have the low mound, small mound, and we have diffuser. The variety diffuser was originally Eutaxia diffuser, reclassified as subspecies of Eutaxia microphylla in 1957, reinstated back to its own species in 2010, a change that practitioners like myself might not have become aware of for many years. Setting up a seed orchard on a slightly different timeline would have diffuser clumped in or split out. And this is a map showing areas that we collect diffuser from, and it's roughly 70 kilometres by 70 kilometres. There's about 250 plants in that area. And the blue dot is 100 of those plants, and they're from a re-veg site, so we don't know the provenance. There is clustered everlasting. We have 3 distinct forms in our region. What we call the type, or standard, which is the grey leaf. We have the fine grey leaf, and we have the fine green leaf. We don't have many of any, and they tend to be isolated plants, rarely clumps. Clustered everlasting has recently, 2016, been divided into 4 subspecies, which I applaud, of which we have 3. So where does that leave the SPA? And then there is gold dust wattle. We clearly have different forms in our region. The Costerman Bible says we can have broadleaf and narrowleaf. Different research papers talk of diploid and tetraploid, and we have suckering or non-suckering, which I don't necessarily buy into. I think it's actually a result of age. Extracts from Broadhurst et al. Read gold dust wattle, and I'll let you read this one. while I get a drink. And this study has revealed that several groups with distinctly quantitative and genetic traits, as well as patterns of emergence and growth, exist within the widely distributed Acacia acanaceae, highlighting our poor understanding of most key revegetation species. There's a lot we still don't know. Actually, the more we know we don't know. So I acknowledge there are splitters and clumpers in the world of species naming, and future testing techniques may lead to an explosion or implosion of species numbers, or probably both simultaneously. When I started, I was told, learn the botanical names. They were more correct and they didn't change. That was just so not true. If a species is a unit of evolution, unable to fertilise or breed outside its unit sustainably, is the species unit something that can be relied upon to keep a species fairly pure when jammed in together in an SPA? Or is that approach opening the door to a myriad of hybrids? In a time of climate crisis and dwindling remnants, what does the SPA practitioner have in the orchard or on the seed bank in storage if the boundaries of species keep changing, splitting or even clumping, as they may or will continue to do? And how can we future-proof? The 15-kilometre rule is long gone for provenance collection. 30 to 50 plants per species in an SPA is long gone. It's way too bottlenecked, and 50 becomes 30. And the Regent Honeyeater Project orchard of 500 per species is a nice try, but 500 becomes 300. And if split into multiple species because the giraffes get in again, becomes 100 or less. Can or should we simply rely on very large numbers of plants per species represented in an SPA? I'm going to suggest for revegetation and bread and butter species, huge numbers in a seed orchard. Let's just say 5,000 plants. 5,000 becomes 3,000, and that's before the giraffes get in. And attrition may also shift the population towards the more resilient gene pool. And to collect from as many provinces as possible within very broad geological question mark boundaries. Pay much less attention to provenance. It would all be mixed up by pollinators over time. Anyway, there's simply not enough out there to be pedantic about. Besides, I like being inclusive. And I say this after 30 years of paying intimate attention to provenance. Let the species within that species, if any, exclude themselves from crossing in unacceptable numbers. I know that some of these suggestions may alarm or offend some, but I'm trying to put forward the position I find myself in, my perspective. As a volunteer, I'm out on the ground, revegetating now, planting systems that will take some years to become useful, that need to be self-sustaining, not sterile, wanting local seed with a broad genetic diversity as possible. We're using seed from seed orchards set up 5 to 10 years ago, or from seed banks that's been stored, unsure of which species is which or will be changed, in a climate crisis, incorporating climate-matched provenance from hundreds of kilometres away, when we need immediate results. We often have tours of our seed orchard, particularly with university students, and I'll point out some of the issues or mistakes, like the varnish wattle. Are you concerned? Yes, I am. I try not to repeat mistakes, but I always say the greatest mistake is to do nothing while worried about making a mistake. But I am concerned that I'm looking at everything through a colonial lens, trying to impose my white fella perspective on everything. With Regent Honeyeaters, there's about 500 left in Australia. You can see some today if you want. They're up at the Burke's Museum. Oh, thank you. Stuffed as they well could be. One of their other names there is the warty-faced honeyeater. So I could be Andie from the warty-faced honeyeater project, which I kind of like. We did have regents in Lurg recently, in November 2020. It was a pretty exciting time. And here's one feeding in Callistum citrinus endeavour in a rural garden. This plant is genetically identical to a plant selected in the 1970s by Swain's Nursery in Sydney. Each year they claimed 5,000 of these plants, all genetically identical. I know, as I was propagation manager there for many years in the 80s. There was no consideration from this bird about don't consume this nectar. We have to fly 100 kilometres through fragmented landscape to get to this provenance. I find that the conversation between restoration and revegetation gets confusing. So to finish off with, we go back to the start. There are giraffes in the seed orchard again. And I couldn't quite come up with the final line for it, so I put some multiple choice up there for people. I settled on E, all of the above. Thanks very much for listening.
Thanks, Andy. It was great to have a little bit of controversy and some giraffes thrown in for the week. So thanks everyone. Thanks all to all the speakers for joining us today. Before we get into questions, there's just a couple of things I wanted to do. One of them is Amelia's going to come up and we're going to do another Menti poll with everyone. So if you could please get out your phones. We'll get a code up on the screen and we'll do a quick question. While that's getting set up, just another plug for getting the presenters' presentations loaded. And also anyone who is leaving early, please feel free to drop off your name tags at the front desk because we do recycle them from conference to conference, just a way to be a bit more sustainable. All right, so I'll give you a couple of seconds to take your photos, enter the code. Just give us a few more likes if you can. I'll assume they're all for me. All right, getting good. Last couple of seconds. All right. This one's just going to really help us in the future to be able to promote the conference and expand I guess the audience based on some of the conversations we've had. So where did you hear about the APCC13? Are you already a member receiving APC or the newsletter? It's good to see that people are managing to communicate even during the time of lockdowns and COVID. We have had 2 years to promote the conference. Okay, fantastic. A couple of more seconds for this one. Great. Thanks so much, everybody. Okay, so I'd like to open the floor for questions. So we've got questions for all of our presenters today. We've got roving mics again. We've got Brad at the background. I think that might be just the one. We've got one already. Well done, Rachel. You're up first.
Thanks, everyone. They were super thought-provoking. Especially yours, Andy. That was great. That was very interesting and putting lots of ideas to us about how what we get up and say as scientists actually translates into practice. And one thing that I wanted to say was, yes, you get very hung up on the taxonomy of things, but I'm super interested in the functional roles that plants play in the environment. So for me, I, will boldly go out and say, almost think that taxonomy is not nearly as important as making sure that the function that the regent honeyeater requires is available at the sites that you're trying to revegetate. So in terms of nectar provision, nesting sites, that kind of thing. I'd be looking at it from a perspective of if you can't source the right kind of species level seed, can you get a good quality analogue for that species that, you can get lots of potential diversity from across its range and plant that instead, unless you've got really strong proof that there's a really clear link between the species and particular plant species. And if it's just about providing nectar, then I think there's room to wriggle in there in terms of not getting so caught up in the taxonomy of things. And I know that's probably controversial to people as well, so bring it on.
Agreed.
Did you want to respond at all, Andy, or are you just in agreement?
Yeah, no, I agree. That's where I'm coming from.
Great.
We get too tied up.
And just a reminder, we do have Ganesha online, I think. If people have got questions as well, I can't see those screens out there. But I think we do have one online.
Yeah, we do. So a question for you, Mark, from Aisha Farouk from MSB. Says that's a really interesting range of studies, and thanks you for your presentation. She'd like to know if there's any work being done looking at seed microbiomes in relation to restoration or translocation success.
Yep, short answer is yes, and it's just begun. We're looking at, I literally just begun. We just got the funding for it a couple of weeks ago, and we're looking at a few species from Western Australia. It's a project sort of spanning across the country, and we've started to isolate and start sequencing DNA from those seeds now. But yes, that's a very initial stage of what will be a pilot study.
Great. And, yes, I think we've got one over here and one over here. There's Gemma.
Hiya. My question is for Nathan. Well, really, I just wanted to say thank you for doing that work. Temperature gradient plates are a bit intimidating, and it was getting a bit confusing with the analysis of replicates, and so thank you. And I was just wondering, I know you've only just recently published that work, but have you or other people had chance to run any more species and use that analysis framework, and has it proven to be robust?
That's a great question. Thanks, Gemma. Yeah, so the motivation for developing that framework is for looking at temperature and moisture effects on seed germination. So we're running this. We've run this script now through, I think, about 20 or 25 different species. And that's helped us to fine-tune the actual workbook to take into consideration things that we didn't initially consider, like really rapid germinating species. So we had to modify the script a little bit to make sure that the interpolations and the predictions of germination in between your checks doesn't actually overestimate your germination and say that you've got 110% germination. So it's working well so far for a lot of different species, and showing a wide array of different temperatures.
Today I'll be presenting a tool that we have developed in collaboration.
Sorry, Nathan.
That's okay. It is a really good tool, so thank you. And yeah, so the germination metric script that I spoke about at the start, that can be easily adapted to for not just the thermogradient plate, but also for incubator-based experiments too. It just requires a little bit of changing of the code for different factors depending on what you're looking at for your experiments. So yeah, things are looking good so far.
Okay, I did say we'd go to one more in the audience, but we've also got one waiting online as well after that one.
Yeah, thank you. I just wanted to say a few words in defence of taxonomy. I really appreciate what your point you were making there, Andie, and I provide mechanisms to deal with uncertainty. And certainly, as a taxonomist, we deal with uncertainty all the time. But as scientists, what we do is we make decisions based upon the data, and it's akin to sort of saying, "Oh, seed storage now, it's so complicated because we've learnt more about seed storage. I don't like it." And with taxonomy, we learn more about the biological diversity that's in Australia, which is just amazing. And we reflect that with name changes. And so I fully appreciate where you're coming from. But again, it's something I think we don't want to take a branch of science and say, "Oh, it's inconvenient, so I'm going to ignore it," because it informs so much of what we do in terms of restoration. We don't want to be planting mixed ploidy populations and generating triploids that are sterile in our restoration projects. So we need to be aware of all these things, and it is complicated. So yeah, thanks, Andie.
I appreciate where you're coming from and broadly agree. But I'd like to get back to the comment, the difference between restoration and revegetation. And restoration is enhancing habitat. And when we've got species that are on the edge and all the things I listed with regarding potential decline, revegetation is creating habitat. And I see they seem to get sort of lumped into the same category, or one wants to be pushed into the other. But I see them as very similar yet extremely different. And that it's the revegetation component that struggles with this sort of restoration overlay of needing to get the naming and everything correct. Whereas it's just a really valuable tool, and it can be quite broad in its approach.
I think we've got a question online, Amelia.
Yes, I've got a question from David Carr online to Ganesha. So Ganesha, did you have a chance to look at the role of birds in spreading and breaking dormancy in wilga?
Not particularly birds, but we've been doing some pollination experiments. So when we were doing that experiment, we haven't actually noticed any particular animal species involved with this, like pollination or seed dispersal in the field. But in terms of seed dispersal, we haven't looked at any animal involvement in this study. So that would be interesting to see whether there's any relationship to dormancy breaking.
Thanks, Ganesha. Have we got any more questions? Yeah, down the front here, Brad. Oh, if you could just wait for the microphone so that online can hear. Thanks.
I'd just like to further talk about the debate about using, mixing up taxonomy. Give an example I came across on Eyre Peninsula where someone had planted Eucalyptus leucoxylon into areas containing what was Eucalyptus leucoxylon, but now has been split off to made into a new species, Eucalyptus pigularis. And what I found there, where the leucoxylon they'd planted was actually seeding, whereas the pigularis wasn't. So there was a real risk of that local, what became a new species, what was leucoxylon, getting out-competed by something brought in. So that just because it's not, it might have just been an amenity planting, still there is that risk of swamping local genetic gene pools and even local species.
Thanks, Richard. I've got Maurizio and then Tony. Over this way, Brad, behind here. So I think these will probably be the last two questions.
Just one last comment on the whole taxonomy thing. I completely agree. That's the reality of science. We can't shy away from it. It's true it's complicated. There are some species groups that are always going to be complicated. Eucalypts are always going to be a nightmare. But we can't shy away from it, and there are ways to resolve it. The other important thing to consider is what you said between restoration and revegetation. Well, I think that's a bit of a simplification. I mean, you can't justify just doing work in an area just for one species, and it's a false economy anyway. You just get the example, for example, of, I think it was Calatris citrinus. Now, if you're planting 5,000 Calatris citrinus from one clone, most likely they're never going to regenerate. They might, but they're not. So you are going to have to replant them again. You might not care because it's not going to be in your lifetime, but have you actually achieved what you were setting up to do as far as re-establishing habitat that is suitable for your target? So anyway, I completely agree with you. It's a problem. It's something we need to think about, but there are ways to deal with it. That's what I'm trying to say.
Can I just clarify? I'm not advocating planting imported species. It's just amusing to observe that threatened species are not bothered by what they're using.
Thanks, Brad. Tony, over here.
Thanks. I have a question for Karen. Thanks, Karen. My question is, does the rate of freezing or warming affect the sort of results you're getting, or does that sort of independent, or can you control that, or is that within your control at all?
Actually, we just have a fancy new machine that will let us control the rate of freezing, and in the differential scanning calorimeter you can specify what the freezing and warming rate is. But for the dry seeds, it's not really so much of an issue, I don't think, but it is something that we could have a look at down the track.
We are at time for morning tea, so unless there's one last burning question, I will be generous. No? Okay, so we've got some gifts here for the speakers. Thank you very much. Anyone who spoke yesterday, please pop out and see Jo, because she does have a gift for you as well. We just didn't quite get around to it yesterday. We do appreciate your time. Thank you very much. Morning tea is served, and after morning tea, we'll be continuing with this theme, and Karen will be chairing the session. Thank you.